What is quantum biology? A good question. I wondered about that too when I was invited to this conference about quantum biology, and I was induced to go partly because I was curious about what people might mean by that. Let me try and explain, and it's a little bit confusing. In physics, for example, one thinks about classical physics and one thinks about quantum physics. That was a division that kind of started 100 years ago with the development of quantum mechanics. And in general, quantum physics tends to be about very small kinds of things like atoms and below, so to speak. And classical physics is kind of what governs the behavior of bigger systems. So what characterizes quantum behavior is, well, partly just they're different kinds of things. So one can say, the word quantum refers to kind of the idea that things are discrete, like the spins of particles are discrete, the energy levels of things are discrete, and so on. That's what the sort of term quantum in its rawest form means. In practice, kind of the way that one thinks about sort of behavior of systems is a bit different between classical physics and quantum physics. In classical physics, for example, when one thinks about the motion of something, there's a definite law of motion. And when you throw the ball, it follows a definite trajectory that is determined by its laws of motion, which depend on gravity, drag if it's in air, things like this. In quantum mechanics, sort of the key idea as it's emerged over the last, 50 years or so, is that things don't follow definite trajectories. They follow a whole cluster of possible trajectories. And what we measure is some probabilistic average of those trajectories. So in other words, in classical physics, definite things happen. In quantum physics, there is this sort of possibility of many things happening. And then there's this kind of tricky business of saying, well, what do we actually measure? What is it about those many things that can happen that we actually perceive? And that's a complicated story that has confused things about quantum physics for the last basically 100 years. I think in our models of physics from the last few years, we have a much better understanding of what quantum mechanics really is, and that it really is associated with many branches of possible history for the universe, both the branching of those paths of history and the merging of those paths of history. That would take us a bit far afield to talk about that kind of thing, but I think we're getting a better conceptual understanding of this difference between classical physics and quantum physics. Classical physics, definite things happen. Quantum physics, there are these many paths of possibility, and some aggregate or average or something of those paths is what we perceive. Okay, so how does this relate to biology? So what one might have thought is that one could say, well, in biology, should we think of definite things as happening, or should we think of sort of the raw material of biology at a molecular scale as having cases where it matters that there are sort of many paths of possibility? Well, let's talk for a moment about chemistry before we get to biology. If we say, what's the structure of a water molecule, let's say, it's an oxygen atom and two hydrogen atoms that are at a certain angle from the oxygen atom. And you kind of have this picture of a water molecule consisting of the oxygen with two bonds coming out of it, and those bonds are attached to hydrogen atoms. And it's kind of a picture where there's a definite water molecule and a definite orientation. And the water molecule, if it's in a microwave oven, the water molecule is kind of spinning around because it's absorbing microwaves and getting lots of energy to spin around from that. Or the water molecule might have might be vibrating with the hydrogens moving backwards and forwards. And that's important in absorbing energy at different from water vapor in the atmosphere and things like this. But the picture is really there are these definite, atoms in definite places with bonds that can be thought of as a bit like springs, and things sort of bounce back and forth that way. That's a very classical picture of a water molecule. Is that a correct picture of a water molecule? Well, for many purposes, it's a quite adequate picture of a water molecule. But if you really want to know what's a water molecule really like, you have to use quantum mechanics, and you have to start thinking about kind of thinking about things in terms of these sort of many paths of history and so on. The thing that you can do when you think about these many paths of history is you can say, what's the quantum amplitude for things? Which means instead of saying there's a definite position, let's take the example of a single of an atom of hydrogen, for example. There's a nucleus, in the simplest case, that's a single proton. There's an electron that's somehow somewhere near the nucleus. And the sort of very early pictures from more than 100 years ago now were that the electron was sort of orbiting around the nucleus, like a little tiny solar system. But what became clear when quantum mechanics arrived was that that wasn't really the right picture, that the right picture is that that electron, instead of following sort of a definite trajectory around the nucleus, it's kind of following a whole collection of possible trajectories. And you can represent that collection of possible trajectories by saying there's a certain quantum amplitude that will turn into a probability. It isn't quite a probability yet, but a certain quantum amplitude for the electron to be found at a particular position relative to the proton at the center. And so instead of saying there's an electron, it's going around in this definite orbit, it's at a definite position at every moment of time, you say there's a certain amplitude for it to be at different positions relative to the nucleus. And so then in the case of something like a hydrogen atom, there are a sequence of different energy levels that correspond to different patterns of where the electron will be found relative to the nucleus. Those in chemistry are so-called orbitals. And again, some of the terminology is a bit confusing, but the kind of lowest energy orbital is one where the electron is found pretty close to the proton, but it has equal amplitudes to be in all different directions relative to the proton. Then you get, that's the so-called s orbital. The names of the orbitals are they're named after German words. So somewhat confusingly, it's the s orbital is the lowest one, then p, d, f. Those are the sequence of orbitals. So the first one, the s orbital, lowest energy orbital, lowest energy possible state for the electron. The electron is sort of can be found with equal amplitude anywhere in any direction from the proton. It has a certain distance from the proton, and its average distance is a thing called the Bohr radius, which is a sort of characteristic length, about 10 to the minus 10 meters, of relative to the proton at the center. The proton at the center is 100,000 times smaller than that sort of the average distance at which the electron is found. But so then, so that's kind of the lowest energy configuration. Then the next configuration is the p orbital, and the p orbital, there are three different p orbitals, and they have sort of lobes that come out going sort of up and down they're in two different directions. And those in that situation, the electron is more likely to be found sort of in the up direction, and sometimes further from the nucleus than that Bohr radius, and sometimes in the down direction. But there are several different distinct p orbitals, three of them, that correspond to those the electron being in different orientations relative to the nucleus. Okay, so let's not dive down into explaining sort of all of quantum mechanics here, but the basic point is that when we think about a single atom, we're thinking about not the electron as an indefinite position, but rather the electron can be has this amplitude to be in different possible positions, and the quantum aspect of it, the original quantum aspect of it is that there are a discrete set of possible kind of levels, like the s orbital, the spherically symmetric one, the p orbital, the one which has sort of the dipole character, and so on, and that there are a discrete set of possible configurations that the electron can be, possible distributions that the electron can find itself in. Okay, so that's for a single atom. the electron is not in definite positions, it's sort of smudged out. Okay, what about for molecules? Well, if you have, for example, a hydrogen molecule, H2, two hydrogen atoms that are, and you might describe that in chemistry as being bound together by a covalent bond. And you might draw that, or you might make your sort of ball and stick model of it as having two balls for each of the hydrogen atoms, and then a stick that is the bond. That's kind of the classical picture of a hydrogen molecule. Well, you can work out the quantum mechanical version of that. There isn't really there's no bond in the middle. That's just an overlap of possible electron positions. And the sort of full picture of the hydrogen molecule says there's a certain amplitude for, there'll be, in this case of hydrogen, each hydrogen atom has one electron. So together they have two electrons. The full quantum mechanical picture has a certain amplitude, a joint amplitude for each of those electrons to be found in different places. Well, the hydrogen molecule is a little bit tricky to do, but you can solve the hydrogen molecule in quantum mechanics. You can find the amplitude for each of those electrons to be in each different position, and they're correlated. So you have to get the amplitude for one electron to be in this position, another electron to be in that position. It is an approximation to that to say that there's just this sort of bond that connects the two hydrogen atoms together. But so the full kind of quantum chemistry version of this would say, well, actually, it's not that these two atoms are a certain fixed distance apart according to the bond. Everything has quantum amplitudes. There's quantum amplitudes to find electrons in different places, to find protons in different places, and so on. That's the full kind of quantum chemistry version of things. The way that one computes that is using the Schrodinger equation. That's a partial differential equation that tells one for the so-called wave function or quantum amplitude, it's that as a function of the position of all of the different elements, the electrons. Usually you only think about, in the simplest case of quantum chemistry, you assume that the nuclei are kind of fixed because they're a lot more massive than the electron. I mean, to give a sense of that, a proton is 1836 times, 1800 times more massive than an electron. So it's so you can, when the electrons are sort of whizzing around and doing different things, the nuclei are much more sluggishly hanging out there. It's a so-called Born-Oppenheimer approximation of quantum mechanics. Assumes that the nuclei just kind of stay put, and it's the electrons that are kind of having all the action, so to speak. But in any case, in using the Schrodinger equation with considerable computational effort, you can solve the Schrodinger equation for something like the hydrogen molecule. Well, so can you do that? It gets more and more complicated, more and more kind of variables in the partial differential equation and so on, more and more difficult to kind of do the numerical computation. There's an additional complexity, which is that electrons obey the exclusion principle. They're so-called fermions. That means that sort of two electrons can't be in the same state at the same time, and that is reflected in the case of the equation in this operation of anti-symmetrization, which is kind of a constraint on how the solutions to the equation can look, and that creates even more difficulty in actually solving these equations either in analytical form or numerically on a computer. So. Or analytically on a computer for that matter. But in any case, the main point is you can represent, you can do, in principle, quantum chemistry, where instead of balls and sticks and so on for atoms and bonds and things like this, you're actually taking account of all these different amplitudes for all these electrons and other things to be in different places. Quantum chemistry is pretty hard. And if you just sort of say, well, I've got this whole collection of atoms, how will it fit together to make a molecule? In principle, you can work that out using the equations of quantum mechanics and so on. In practice, that's not really possible for molecules with more than at most tens of atoms. And even at tens of atoms, it's really hard. So the full quantum picture of how all these things fit together, really difficult to do. It's also very confusing because for something like a water molecule, the one of the things, I mean, this is a sort of deep can of worms. Not okay. So I mentioned that with a hydrogen atom, that the lowest energy configuration of a hydrogen atom has the electron with equal amplitude in all different directions. It's a spherically symmetric orbital. Okay, well, it's a general rule about how quantum mechanics works that the lowest energy configuration of systems tends to have that spherical symmetry property. So if that's the case, how can we have a water molecule where we say there's a water molecule with definite positions for the hydrogen atoms, and it's a water molecule that has this orientation? Why isn't it the case that the sort of lowest energy version of a water molecule is a water molecule that's kind of where all directions are equal, and you can't really say that it's an oxygen with hydrogen sticking out in a definite orientation? This is a tricky point, and it has to do with one of the many pieces of messiness in quantum mechanics. And okay, so in the case of an electron in a hydrogen atom, you say, well, there's a quantum amplitude to find the electron in different positions relative to the nucleus, and all different directions have equal amplitude. But if you actually, quote, measure where the electron is, then you can come to the conclusion, yeah, the electron was in this definite place. In other words, there's this whole bundle of possible histories of the universe, but as soon as you sort of actually observe something, as you'd say, well, actually, I know it was, in fact, it was in this particular, I picked up this particular history of the universe. The way I think one should think about it is that our sort of the universe has all these different branches of history, and we happen to exist in a particular branch of history, and that's the one which we sample. So even though the sort of the equations say the electron has equal amplitude to be in all these different directions, once we've observed where the electron is, then we know the electron was in that definite direction. Okay. So, but the confusing thing is that we start off saying, well, it's in all possible directions. In the case of a water molecule, we might say, well, it could be in all possible orientations. But the fact is, we imagine it using our sort of classical physics thinking to be in this definite orientation that corresponds to our ball and stick model. And the question is, why is that a reasonable thing to do, to think about it in terms of being in a definite orientation? And the rough answer to that is that just as in the case of an electron with a hydrogen atom, we make a measurement on it. We do that by having it go through some magnetic spectrometer or some such other thing, and it has that the atom is led to go either to the left or the right or something like this. We take an action that causes us to effectively make a measurement on it. Well, a water molecule just hanging out near other water molecules, the picture is that all those other water molecules are in a sense taking actions on the first water molecule. In effect, all those other water molecules are sort of continuously measuring the water molecule that we're interested in. And so that means that this thing where we say, well, it's in a definite orientation, is sort of a reasonable thing to do because the environment is somehow continually making measurements on that water molecule. This is a complicated thing, and people, I have to say, the pros often get extremely confused by this issue. But I think the right picture is that it's the sort of the water molecule can be thought of in this sort of classical way with balls and sticks and so on in a good approximation because it is kind of continually being jiggled by other water molecules, which is like sort of an experiment being done, but it's continually happening. The experiment being done that determines where the electron in the hydrogen atom was. Okay, so this was a long story about sort of quantum chemistry, and most of the time when one does chemistry, you use at best semi-classical methods, which typically means, well, you have a molecule, it's more or less balls and sticks, but there's a certain amount of noise. The molecule is sort of being perturbed, and that kind of represents sort of quantum processes, quantum effects. Usually molecules are perturbed by the presence of heat. The temperature is is the kinetic energy, the energy of motion of things. And if you have little molecules like water molecules, for example, they can just be whizzing around and maybe rotating or something like that. If you have a big molecule like a protein, it's a big floppy thing. And maybe a water molecule crashes into it, and then it will make the protein deform in a certain way, and so on. Well, usually kind of the analysis of what happens to the protein molecule is something which is kind of done at the level of more or less balls and springs and sticks and so on. It's a big, let's say you have a protein molecule with with 1 million atoms in it. Well, it's completely infeasible to do quantum chemistry where you're solving the Schrodinger equation for all of those different atoms. Instead, you take a semi-classical approximation where you think of the molecule as being a bunch of masses corresponding to the atoms and a bunch of spring-like things corresponding to the bonds between those atoms. That's just an approximation to what's happening at the true quantum chemistry level. But then you're doing computations on those molecules, and that's already very difficult. Typically, the thing that is sort of the high-tech piece of this is so-called force fields that are the things that determine for two atoms what are the forces between those two atoms. So roughly, for example, if the forces if you had atoms that were charged, ions, then roughly at least at long distances, the forces will be determined by electrostatic forces, and they would be an inverse square law of attraction. Let's say if the molecules were with opposite sign charges. But in fact, the forces between molecules are more complicated. There are very common cases, so-called van der Waals forces, where actually the thing called the Leonard-Jones potential, where at long distances, the force between two neutral atoms, not ions, but atoms where there's a sort of a match between the electric charge of the nucleus and the electric charge on the electrons, neutral atoms roughly attract each other in a way where the force of attraction goes down like 1/r⁶. It's actually sometimes 1/r⁶, sometimes 1/r⁷. Those are so-called van der Waals forces, and neutral atoms will attract each other by those forces. Then at short distances, the atoms kind of start repelling, usually by the 1/r⁴ force law. So roughly, it's 1/r⁶ minus 1/r⁴ is kind of that's the so-called Leonard-Jones potential between two atoms. So sort of the high-tech thing in working out how molecules behave is to have those force fields, those laws of attraction and repulsion between atoms. And the thing I told you is just a rough approximation to kind of your average simple atom. In reality, the force fields are more complicated and depend on which type of atom you're dealing with, and so on. But that's where a bunch of sort of fancy supercomputing gets done, and custom computers get built to try to do the most accurate sort of figuring out of the motion of molecules based on those kinds of force laws. And so a not untypical thing, I mean, the best efforts these days are looking at the motion of molecules on timescales of order femtoseconds. That's timescales of order 1,000 trillionth of a second. That's enough that you kind of see the wiggling of electrons at the level of that at speeds that correspond to you see the kind of timescale of wiggling at frequencies that are not quite the frequencies of light, but at least the frequencies of microwaves and things. And so you're getting sort of this series of configurations of molecules down to the level of what happens every femtosecond. And one of the things that's really tricky about quantum chemistry is that there's motion on many different scales. So little places where there are electrons, or little places where there are hydrogen atoms hanging off the backbone of a protein, for example, those hydrogen atoms will wiggle around on timescales of order femtoseconds, but the full big protein might be sort of moving around and sort of moving like a snake or something on timescales of order microseconds or more, which means that there's you have to be able to sort of deal with when you do computations, you have to be able to have, you know, things where you're taking account of effects where there are factors of a million difference between one effect and another effect. So you have to be able to sort of get the things that are at a millionth scale and the things that are at sort of one scale, and you have to be able to take account of all of those things. So it's very hard work. But as I say, most of what's done, you're not even dealing with full quantum mechanics there. You're just dealing with the semi-classical approximation of the motion of molecules and so on. Okay, so what does this all have to do with quantum biology? Well, one thing that you might wonder is, are there in fact quantum effects that truly depend on sort of these many paths of history and all that kind of thing? Are there quantum effects that are somehow important in biology? For individual molecules, there are certain effects, like the delocalization of electrons that happens in rings of molecules where there is a ring of carbon atoms or something like that. There's some sort of trickiness that is a very quantum mechanical phenomenon that depends on all that quantum chemistry stuff, at least in some approximation. Are there things where sort of what happens in biology really depends on what's happening at the level of all of this sort of complicated quantum stuff? Probably the answer to that is most of the time it doesn't matter. Most of the time molecules are flopping around, and you can use this sort of semi-classical approximation where there might be some sort of quantum noise that's kicking things around, some sort of thing that's sort of an injection of randomness that comes from quantum effects, but that most of the time you can just think of the molecule as being like balls and springs and so on, maybe with these fancy force laws if you want to. But so there's sort of a question of, are there effects which really depend on kind of this many paths of history, quantum mechanics kinds of things, or this discrete energy level aspect of quantum mechanics and so on? Are there features of biology that depend on those things? So that's sort of one question. The answer is it's not clear. I mean, my own guess is that most of the time it doesn't matter. But one feature of biology is that in a sense, biology has an infinite chain of footnotes. When you say it never matters in biology that this, that, or the other, it never matters what the Earth's magnetic field is like, for example, that will not actually be ultimately true. There'll always be some kind of creature that has some weird way of depending on how the Earth's magnetic field works, let's say. So in biology, unlike in physics, where things tend to be rather clear-cut, it's either there is an effect or there isn't an effect. In biology, it's like, well, there usually isn't an effect, but just sometimes there is an effect. So how does this relate to quantum mechanics? Well, there are a few particular phenomena where there are sort of quantum phenomena which seem to be important in biology. So in fact, one I mentioned related to the Earth's magnetic field is that there's a question of, do animals, organisms, folks like us, are we sensitive to this Earth's magnetic field? I mean, if we have a compass that has a magnet, a little tiny magnet suspended somewhere, that magnet will line itself up with the Earth's magnetic field, and you'll be able to tell which way north is, let's say. But the question is there anything in us that does that? Well, we could have little tiny pieces of iron, for example, like little iron filings that would actually move around in response to the Earth's magnetic field, and so that we could have little tiny compasses in some organ in us. That's one possibility. The other possibility is that those tiny compasses could be really, really tiny, because it turns out that atoms and nuclei and electrons and so on, they all act as tiny magnets. And so there's a certain tendency for atoms that have high so-called spin, which is essentially some atoms, some nuclei and some atoms will behave as if they're like little bar magnets that have that can be moved around by a magnetic field, because bar magnets try and align themselves with the magnetic field. One of the possibilities that seems very quantum is that that alignment could be happening at the level of uh, um, individual atoms. So in particular, there's been a long time claim that in certain organisms, particularly pigeons, that there are atoms that are sort of embedded in cages inside proteins, and where those atoms will tend to move around those atoms have essentially magnetic, like little tiny bar magnets, and they move in response to the Earth's magnetic field. And something about that cage is able to detect what the orientation of the atom is relative to that cage. So that's a claim where sort of a thing that's very quantum mechanical, the spin of an atom, is something that has sort of an effect on biology and that causes pigeons to know which way is north type thing. So that's one kind of effect. There are other kinds of effects that people have talked about, which I'm not a big believer in, that relate to kind of the features of cells. So cells have lots of little fibers in them, so-called microtubules, and as well as fibrils of various kinds. But microtubules are often on the surface of cells, and they're little. Think of them as little pieces of fiber, and they can move, and they're just made of protein. But there's a question of whether sort of the pattern of what microtubules do depends on quantum mechanics. It's kind of an attempt to say, well, quantum mechanics might affect how brains work, and the medium for that happening would be through these microtubules moving around different ways. I don't happen to believe that this is really an important effect, but in the general principle that there's an infinite number of footnotes in biology, I'm sure there's some effect there. It's just a question of whether the effect is of any significance or not. Okay, so those are some ideas about how kind of sort of traditional quantum mechanics might have effects in biology. But in fact, what people sometimes mean by quantum biology today isn't really any of that. It's really the interaction of things like light with biology. And why does that even make sense to call it quantum biology? Well, it makes sense because if we're thinking about the interaction of light, if we're thinking of light as consisting of a stream of photons, then photons are the quintessential quanta. When we think about something like light, there's different views of light. We can think about it as kind of like a wave, but we can also think about it as a big cascade of photons. And for some purposes, it's much more useful to think of it as a cascade of photons, because, like, for example, in our eyes, when light falls on our retina, it's individual molecules, photoreceptor molecules that are proteins, that where an individual photon interacts with that photoreceptor and generates a cascade of electrons inside the photoreceptor, and that's how we register that we saw some light. So it's sensible to think about the light that falls on our eyes as being a stream of photons, rather than just saying, well, it's a wave and it's the aggregate of many photons, because the individual photons can have an individual effect on generating electrons inside the photoreceptor cells in our eyes. So in any case, one sort of meaning of quantum biology is it's really about the effect of things like photons on biology and what's the relationship between sort of individual photons and what happens in biology. So it's clear that photons have an effect on the photoreceptors in our eyes. They're specifically generating this cascade of electrical activity when the right frequency of photon falls on the right photoreceptor in our eyes. So there's questions about, well, where else do photons show up in biology? So one thing people claim is that chemical reactions generate photons, and that you might be able to detect those from different things happening in biology. Well, at some level, okay, boy, we're getting into lots of different things. Let's talk for a second about what heat is. So if you take a block of metal and you heat it up, what is different between the hot block of metal and a cold block of metal? Well, the answer is in the hot block of metal, the electrons and atoms in the block of metal are moving around much more than a cold block of metal. The heat is associated with the microscopic motion of molecules and atoms and electrons and so on in the material. So that's sort of heat in a material. But there's also radiant heat. There's and that's corresponds to streams of photons. So when you heat something up, it eventually starts to glow. First it glows red hot, then it glows white hot, then it glows blue hot. And what's happening there? Well, what's happening is that from the motions of all those molecules, those molecules are emitting photons as they move around. What's really happening is that in atoms and molecules, there are this sort of the molecule is in a kind of it gets into this excited state where it has a higher energy. And one way that it can lose that energy, one way it can lose the energy is by crashing into another molecule and just exchanging its energy making its energy, turning its energy, its internal energy into energy of motion of the other molecule or the molecule itself. But another thing that can happen is that energy can be emitted as a photon. And so when it's by the time something is red hot, it's emitting visible light photons. If it isn't quite red hot yet, it still will emit photons that are slightly lower energy than the ones that are perceived by us as red. Those lower energy photons are infrared photons. So when things are at all hot, they're emitting infrared photons. So when a thing that's hot both has motion of molecules inside it and is also radiating photons that are the frequency of the photons depends on how hot the thing is. But if the thing isn't very hot, if it's like, only, I don't know, 100 degrees centigrade or something, it won't glow. It will just emit infrared photons of a certain frequency. And as you increase the temperature, the typical frequency of the photons will increase, going from, for example, when it gets into visible range, red to, well, white is sort of a combination because the distribution of frequencies of photons is quite broad. It's the so-called Planck distribution and or black body distribution. And things are never green hot because it's never the case that you're only generating photons that are in the kind of green band of frequencies. Instead, by the time you would be green hot, you also have a certain amount of red hotness and blue hotness. And so the combination of those things makes it seem like the thing is white hot. But in any case, so you're always emitting infrared photons whenever something is hot. Okay, so what does this mean for biology? Well, in biology, there are lots of chemical reactions that happen, including ones that produce heat. If we humans are regulating our temperature and keeping ourselves at, if you're in the sort of US 98.6 degrees Fahrenheit or whatever, if we're maintaining that temperature, mostly our liver has chemical reactions going on that produce heat that can maintain that temperature. But so it is routine for us in biology to have chemical reactions happening that produce heat. So we're producing infrared photons that way. But the question is, for example, one question is, are there other kinds of processes in biology that aren't sort of obviously just generating heat, but are somehow occasionally generating visible light photons, for example? In other words, is it the case that sort of we all glow in the dark just a bit because of chemical reactions that happen in us that produce a small number of visible light photons? So there are claims that that's the case. Probably, as I say, there's always more footnotes in biology, there probably are some visible light photons that get produced. Just to give you a sense of why something like that might happen, one of the more bizarre phenomena is if you break something, like if you have a piece of, I don't know, a pencil or something, you snap it in two. Okay, well, it makes a noise as you snap it in two, but as you snap it in two, it actually generates some okay. What's happening when you snap it in two? Well, you're breaking a bunch of bonds between different atoms and molecules and so on. And in fact, when you break something, some of what gets broken are actual bonds inside molecules and so on. And there's a fair amount of energy associated with those bonds. And when you break them, that energy is released. And so when you break something, there will be a tiny rate of production of visible light, even X-rays, as certain chemical bonds get broken, because you took, I mean, even though the chemical bonds are quite strong, they're not strong relative to the amount of force that you're putting onto the pencil to break it. Each individual bond is absolutely a microscopic energy compared to what you're putting into the pencil to break the whole thing. And that breaking action that you make, somewhere down there, there'll be at least a few actual chemical bonds that get broken. And the result of breaking those bonds is to produce photons that have quite high energy. So you actually produce, in breaking things, actually produce visible light, tiny amount, even tiny amount of X-rays. So it's probably unsurprising that in biological reactions, biological chemical reactions, that there's a small rate of producing, for example, visible light and so on. Is it measurable? Is it important? Is it diagnostically important? I don't think one knows yet. But that's an example of something that you could think of as being a quantum biological phenomenon, because it sort of involves quanta, like photons. It's a little bit of a stretch, because you might think that when you're saying quantum biology, you mean kind of the quantum chemistry thing, but that isn't an example of that. And so you can ask a bunch of questions about whether, what effect, for example, things related to photons have on biological organisms. You know, how much does light, how much effect does light have on things? How much effect does magnetic fields, do magnetic fields have on things? I think one of the things that has become sort of interesting in recent times is that, well, one thing is, how far does light get when you shine it into you? So, you know, you take a flashlight, you put it on one side of your hand, well, you see some light coming through the other side of your hand. It typically looks very red. It isn't red because there's blood in your hand or something like this. It's red for the same reason that sunsets look red. It's red because if you look at white light, which consists of all different possible colors of light, it turns out that blue light is more easily absorbed by things than red light. And so, for example, when you see a sunset, you're seeing what's left over, that the sun looks red because the blue light from the sun is producing both red light and blue light and other colors of light, but the blue light is kind of scattered away. It's absorbed in the it's basically, well, it's basically absorbed by the air. It's not quite absorbed, it's actually scattered in a different direction, which is why the sky looks blue, because you're getting light that came from the sun, but it's being, the blue light, more than the red light, is being scattered in directions away from the sun. But when you're looking straight at the sun, what you see is, well, in the case when the sun is high in the sky, you're just seeing kind of the lots of frequencies of light. When the sun is kind of close to the horizon, you're seeing it through a bigger column of air, and so what happens is more of the blue light is removed, and so the red light is left over. It's basically the same phenomenon when you have a flashlight shining through your hand, that what gets left over is the red light. So there's a question of how, if you have intense light, how deep does it get when you shine it into you? And so one of the things that's pretty clear is that, yes, you can sort of shine light through the, you know, if you shine, a little bit of light will get through your skull and get into your brain and so on. And does that have an effect? Well, I don't think one really knows. There is, you know, even things like the pituitary gland that sits in the middle of the brain. It's known that has a lot to do with how your circadian rhythm is set. It secretes chemicals depending on whether you're, you know, in sunlight or not. Is there a direct effect of actual light that falls on your head, so to speak, a little bit of it getting into the pituitary gland and having an effect on how it produces chemicals? I don't think one knows at this point. But another example of sort of a quantum biological phenomenon, at least you could call it that, is the effect of light on direct effect of light on biological tissue. There's also the question of whether magnetic fields have an effect on biological tissue. If you go get an MRI, there's a strong magnetic field that's used to actually, it makes use of the fact that there is that hydrogen atoms can get sort of lined up in the direction of the magnetic field, or if you have contrast agents like gadolinium happens to be something where the nucleus of gadolinium is something which has a high spin, and so it has a sort of high magnetic moment. It tends to line up with magnetic fields. But in any case, the way that MRI works, it relies on having a high magnetic field. And so if you get an MRI done, it's kind of, so for all we know, what appears to be the case is that we just don't notice that magnetic field. Nothing, it has no effect on us. It has an effect on the hydrogen atoms in us, and that effect can be sensed by looking at the emission of microwave radiation from those hydrogen atoms and so on. But as far as us as biology, it doesn't seem to have any effect. Is it really true that magnetic fields have no effect on us? Well, there's some evidence that it does have some effect. As I mentioned, for pigeons, it's believed that there are particular kind of structures where the Earth's magnetic field, which is quite weak, very weak, it's 100,000 times weaker than the magnetic field in a typical MRI, that the Earth's magnetic field can be sensed by a pigeon. An interesting question I don't know the answer to is what happens if you put a pigeon in an MRI machine? I wonder what the answer to that is. I don't know. It may be, as is often the case with these sensory organs, that if it's sufficiently overloaded, the brain just doesn't sort of register anything that's happening. But there are definitely questions about that. In any case, there are sort of these kinds of phenomena that are sort of the direct effect of things about photons, electromagnetic fields, and so on biology. I mean, there are claims that that there are all kinds of different things related to electrical and magnetic phenomena affecting biology. I mean, you can ask, if you use a small electric current, does that small electric current have a substantial effect? You can certainly, if you have a larger electric current, you can have effect on muscles. You can, that electric current will make muscles contract, things like this. But will a small electric current have an effect? Will it sort of reset certain things that happen in the reactions that are going on, or something, or some feature of how the cells in you are set up? There's like I say, everything in biology has footnotes, and so there's surely some effect. How big the effect is it an effect that's important for medical purposes? I don't think one knows. I mean, there are a lot of funky things. I mean, one feature of biological organisms is, you think of them as having separated pieces. You've got the brain that's separate from the gut, that's separate from muscles, and so on. But in the end, there are sort of effects that go between all these things. So one of the things that's emerged in recent years is the kind of the gut-brain axis, it's usually called, where features of the microbiome, the population of microorganisms in your gut that sort of are involved in the digestion of food and so on, those that the sort of the state of your microbiome can have an effect on your brain, can have an effect on it, it can have an effect on your brain. And that's sort of surprising until you realize that there are nerves that go both from the brain to the gut and from the gut to the brain. And exactly how those things work is still quite unclear, but that there is a connection seems to be the case. And another one of those connections is between the immune system and the brain. you might have thought your immune system making antibodies, having T cells going and attacking things and so on, perhaps having autoimmunity where you're attacking your own cells and so on, that all those things were just features of the cells in the immune system and that there was no brain control to all those things. But it's becoming clear that there are ways in which at least nerves are connected to things in the immune system. There are connections between sort of the brain and the immune system. Now, can there be connections to what you kind of consciously think and the immune system? Quite possibly. I mean, once there's a connection between the brain and the immune system, it's there's a kind of very fuzzy line between what you consciously think and all the things that are going on in your mind, in your brain. So, but it's all very subtle because they're often very small effects where in principle you can measure some effect, but does it matter or does it not matter? I mean, at some level, everything matters. I mean, if you eat one piece of chocolate versus one piece of fruit or something, that will matter to some microorganisms in your gut. Probably 10,000 microorganisms are affected by that one piece of chocolate versus that one piece of fruit. But do the fact that those 10,000 microorganisms end up doing this versus that, does it have an actual consequence to you that's of any perceptible importance in sort of your medical history or whatever? One doesn't know. I think the thing that even confounds things even more is when you're thinking about, what has a medical effect? Well, as I mentioned, the brain affects things in the body, from the immune system to the things probably about the microbiome to all kinds of other things. Things that we think of as not being under conscious control actually are to some extent under conscious control. I mean, I don't know, you can end up, if you have enough of a sort of a shock of, something sufficiently surprising happens, you can miss a beat with your heart, so to speak. And that's, although the operation of the heart is governed by pieces of the brain that are not directly connected to your thinking, that there are claims that some people can mediate their heart rate just by thinking, but most of us can't do that. Most of us, if we run upstairs or something, our heart rate will go up, but that's for reasons that have nothing to do with us thinking about running upstairs, or at least we don't imagine so. It has to do with the sort of lower level action of our brains. So in any case, the idea that the brain can have an effect on things which we usually think of as just humming along without sort of conscious brain control is something that undoubtedly is the case. I mean, take breathing, for example. Most of the time, we don't think about our breathing. But if we put effort into it, we can think about it and we can hold our breath or do whatever else. But most of the time, it's kind of an autopilot. And things like our heart really are much more on autopilot, and it takes much more. And I don't think most people don't know how to, by sort of consciously concentrating, change what happens with their hearts, even though they can change what happens with their breathing. So, the question is then, so there's an effect of what you think on other things in your body. And then that leads to all kinds of difficult sort of confounding features when you try and do medical experiments. So one feature is the placebo effect, where if people are convinced enough that something is going to make them better, it's that through mechanisms that aren't really very, very well understood, that can mean that their brain is sort of sending the signal, hey, this should make me better. And so maybe it's affecting the gut, maybe it's affecting something about some muscles or something like this. And by golly, it makes you better, even though the actual, if you were doing a clinical trial of a drug and you get the placebo, that means you get something which is supposedly inert rather than getting the active drug. And so, but even the placebo, from the idea that you're getting this thing, you don't know whether it's a drug or the placebo, that idea can make you better, so to speak, without any actual sort of medical molecular thing kind of happening. So I have to say, it's a funny thing. I was at this conference, I listened to a few presentations, and some of them were very, there's this tiny effect and we only see it sometimes, and you have to be in exactly the right place, and it matters whether it's this practitioner or that practitioner that's doing the thing and so on. And, I'm thinking to myself, if this was kind of shamanistic medicine, kind of the witch doctoring and so on, would the presentation really sound much different? But then you realize that there's this complicated mixture of kind of what you think is going on versus what is molecularly going on and so on. And so it's very hard to disentangle those effects. And particularly in lots of kinds of medical things, sort of the ultimate effect is quite small, though it may have a big effect on you. You have a little bit of I don't know, you have, your toe is hurting. And so the result of that is that you limp slightly. And then the result of you limping slightly is that you kind of cause, your back to get thrown out in some ways. So the small effect can turn into a big effect. And so, just a little bit of a sort of tweaking of whether you're believing in something, not believing in something, whether there's a molecular effect or not, can end up sort of spiraling out to something that is a notable effect. All right, that was a super long answer to that question about quantum biology.